All right, now here's the interesting. We have this last video, we're going to talk about why, if you look at a bacterial genome, that didn't work. There we go. That you're going to find more than one lac operon in your typical bacteria. You're going to have a lac operon over here, and have one over here and over here, and they're all going to perhaps work. Maybe some of them don't work, though. We're going to talk about why they exist at all. Because it doesn't make sense. Seems like that's a waste of space in your genome. And the bacteria actually are limited on how many things they can fit into their genome. This little loop is not infinite, can't get infinitely large. It'll break. And unlike a chromosome. So this little loop has a limited amount of genes it can put in there. Why would you want to waste 3 of these? And the average bacteria does have 3. Why would you have 3 of these lac operons in there? Well, the reason is that we find that most of these operons are very similar to the lac operon, and they have most of the same structures. All right, Tata. And they'll have a few genes. Maybe they have more, maybe they have fewer. So our lac operon has 3. Or I don't know if you call them genes. Open reading frames is what we should call them. Protein coding sequences. But the lac operon, maybe this other operon has, A, B, C, and D. And we look at these and we do studies just like we've done with GPX, and what you'll find is that there's a whole lot of sequence similarity between these. And it's thought that these other operons that have other functions probably evolved from something like the lac operon. Maybe there was an original operon millions of years ago that did something, but it has slowly turned into all these other operons. And one of the original ones was the lac operon. It's pretty clear. But the idea of why do you have multiple copies? And in particular, why? Why would you waste that space and potential energy to build it and maintain it and stuff? Well, it's mainly because of the whole idea is the cell evolves, and it needs a way to, let's just say, play around, experiment. I guess that's a better way to talk about it, with the functionality of these proteins. What we find is that when you take especially Z and you change the code a little bit, not much, but a little bit, that it will stop digesting lactose, but it may start digesting a different sugar into glucose and something else. In other words, what we find is the function of these, the lac operon digests lactose, but one of these others may process some other chemical, destroy it, turn it into something else. And if it happens to turn it into something more useful, hey, that sequence is going to hang around, all right? That's going to be a conserved sequence. And so by having multiple operons, multiple lac operons or other operons that do a particular function, they can mutate and they can be free to change without killing the cell. In other words, if you've got 3 of them and 3 lac operons and you start changing one of them, well, you've got 2 backups, all right? So you've got a backup in case it's mutated. But this backup that mutated, well, it's not going to just disappear overnight. It's going to change ever so slowly, and it may change into something completely worthless. It may change into something that's deadly and kill the cell. But every now and then, 1 in a million, it changes into something that gives the cell a new function. And if it does, then suddenly that one will do what we call fixate. It will fix, and it will become a permanent gene. And then it'll go and have its own duplicates all right, later on, and go through the same process as the lac operon does. But when one of these changes enough and it actually produces a positive effect for the bacteria, then it will be more likely to hang around, and it will, even if it's not necessarily harmful, if it's just not, I mean, if it's helpful, it's just not harmful, it'll probably stick around for a long, long time. And that gives it even more time to try and find a functional purpose. And so you get a lot of these in the genome of these bacteria. And yeah, it's a wasted space, but it's also a way for the bacteria to adapt to its environment, for it to develop abilities that it didn't have before, and ultimately maybe become a eukaryote or something like us and evolve in the future. But it takes a long time for all of this to happen. Now there is, we need to learn the how these various lac operons operate together. So this is something I did not cover in class, so this will be new even for the people who are in class on Wednesday. So let's say we have 3 operons. We're going to call it lac 1, lac 2, and lac 3. And they have all the normal parts, at least the basic parts. All right. So we've got 3 of these in there, and we also have the I1, I2, and I3. Just trying to get through this real quick. And a tata. 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 So let's think about what happens when a mutation occurs. Right now you've got 3 functional proteins. But let's say, let's say something gets altered. Let's say you mutate, there's a mutation. And the mutation, let's start with a major mutation. You get a change in the O protein, and that sequence changes. So if that sequence changes, what's going to happen? What happens? Well, is made, and what does it do? It likes to stick here, but it likes to stick to just that sequence. If you get a mutation there, what happens? It can't stick. And if it can't stick, what does that mean about this particular protein? This particular operon? It means, what is the normal function of this? It's to turn it off. That means this one will be always on. All right, and that's not a good thing because you're going to be wasting resources. Always on is not a good thing. A mutation right there would lead to all kinds of wastes of energy, and probably this cell, if it wasn't living in a good condition or in milk or something, it's probably going to die because it's going to run out of energy. It's not sustainable. All right. Well, let's undo that. I should learn to erase. Oh, definitely should learn to erase. I'll do it this way for now. erasing all of this. Oh, that's going to take forever. I won't do this again, I promise. Okay, we're almost done here. Okay, oh, I got to get rid of that X there. Okay, so let's do a different scenario, and let's mutate I so that it doesn't work. So what if this I doesn't work here? All right, so that I is not working. Well, what is its job? Its job is also to stick here and turn this off. So without the I, you would go, oh, well, that's not going to work. The I isn't made, so you're not going to stop this from being on. It's going to be on all the time. That's where you would be wrong, because look down here. You have two other I's, the ones that went with the other operons. Now, assuming they got duplicated as well, then these down here, these I's, they produce more than enough. Remember, this is a cheap protein to make. They make more than enough, probably, to cover their two operons and this one. And because you don't need it, you bind two here, and maybe two others at one of the other operons, and should still work. And in fact, that's exactly what we see with experimental results. We mutate one, and everything still operates just as it normally would. You see really no difference in how the cell operates, how much it produces, nothing. All right, so that's an interesting idea, is that the I, one of the I's getting mutated, doesn't affect any of the three. All right, now what if you mutated a Z? Okay, so this Z doesn't work anymore. Well, no problem. You've got two backup copies. You still make Z. Everything maybe it doesn't work as efficiently as it did before, but it still works. All right. What about if you mutated a Tata box? Well, a Tata box, if you were to mutate a Tata box, what? Nothing is ever made. That's like a permanent off. There's just no way to recover from that. All right. Now, if you only had one I, all right, now that would make some difference here. If neither of these, if you only had one I to operate with all three of these, then any changes to I, it's going to alter all three. All right. Now, I just want you to realize how this works. And if you mutate some of these, say you mutate this one as well, okay, it starts making some strange things, but this one also is making something that's wrong. But ultimately, you still got one that's functioning. And technically, even if this one didn't exist, these two are working. These two are working as long as there's one functioning I. These two could work in conjunction, and things would probably work just fine in that cell. Plus, these two could be experimenting, right? They could be experimenting with digesting something else, all right, or detoxifying something else. So these two working together could be developing the ability to, maybe this one is learning to slowly, giving an advantage to digest some other sugar, and this one is slowly evolving to detoxify something that it didn't detoxify before. So keep this in mind. How these operate together is very important, and why you have these three is if you ever take a class in bacterial genetics, they will take the lac operon and they will ask you how this all works together. This is about all I'm going to ask you for, but we might do an exercise where you have to go through and figure out which of these is working or something like that. But I wanted you to understand how the evolution of these work and how you go from one of these, all right, to some new operon that has a different function.